Hi, uh, my topic is discovery of antiviral gene targets based on metabolic modeling. So this is introduction part. This shows how virus infect and reproduce themselves and release to the outside of the host cell. So usually antivirus uh, drugs target the first step, which is biosynthesis. For example, remdesivir, one of the COVID-19 uh, drug, it degrades into this molecule, which is really similar to adenosine. So this molecule is used instead of adenosine during the biosynthesis, the RNA replication process. So it can uh, resist to the virus reproduction. So I also focused on the same step, biosynthesis, but my perspective is a little bit different. The host cell's metabolism is altered by uh, virus infection. So if we can revert this altered metabolism to the normal state, this can be a new antivirus uh, uh, strategy. So this is uh, overall scheme of my research. First, we extract GEM from four data sets, and we apply gene knockout simulation and compare fluxome with mock sample using UMAP. So we can find which genes knockout can restore the metabolism similar to the mock state. So these are four data sets which uh, has the human lung cell lines with the human coronaviruses, and we extracted two gems from each data set, one for mock and one for infected. In these two cases, in GSE 186460 and GSE 198388, 8398. In these two data sets, infected sample and mock samples are in the same UMAP cluster, which means um, their metabolism is already similar to each other, which means uh, the uh, the metabolism alteration by the virus infection is not big enough for the analysis. However, in this case, GSE 193.169, we can clearly find gene targets. So when the genes listed in here, the genes involved in oxidative phosphorylations are knocked down or knocked out, the metabolic state goes to similar to the mock state. And in this case, in GSE 155986, when the genes involved in the opioid metabolisms are knocked out, the metabolism goes back similar to the mock state. So from here is an experimental validation part. The gene targets in GSE 193169, the gene targets are involved in the oxidative phosphorylation, and specifically the genes encode the cytochrome C oxidase, which is a complex 4 in the oxidative phosphorylation pathway. So we used ADDA5, which is a small molecule inhibitor of the complex 4, and we tested by RT-qPCR and the plaque assay to show the viral gene decreases by treating ADDA5. And in this case, the genes related to the porphyrin metabolism, which shows two genes, SLC25A38 and ALAD, which locate at the early stage of the ham generation pathway. And unfortunately, there is no small molecule inhibitors, so we knock down with the shRNA. And also, we used RT-qPCR, and in this case, we used the TCID50 test to show that the viral gene decreases by these two shRNAs. So this is conclusion of my research. Antiviral gene targets identified by the combination of the gene generation and single gene knockout simulation and UMAP analysis were experimentally validated. And with this process, antiviral gene targets can be identified in a very short time. So gem construction about few hours, and gene knockout simulation also few hours, and UMAP analysis is done in few minutes. So we can find a gene targets within a day. And interestingly, in GSE 193.169 dataset, target genes were downregulated in the infected samples. Knockout of these downregulated genes showed antiviral effect. It's quite interesting. So this research can be a novel approach for the antivirus strategy. Thank you for listening.